So we were tasked with the job of talking about transcriptional regulation and some of the issues associated with this. So I think we spent a lot of time, at least in our session, talking about our long-term vision. So if you can build complex networks, whether out of transcriptional regulation or others, we'd like to be able to tackle some really big grand challenges. And some of these came out to be, for example, personalized human therapies. So instead of perhaps thinking of one circuit that's going to treat every variant of a particular disease, could we actually pretty rapidly, after, for example, profiling, a particular dysfunction in a set of cells, design a transcriptional circuit very rapidly to meet and to bring that disease circuit back to homeostasis. So that was one of the kind of long-term visions that people would like to go towards. People would also like to, I think we talked about being able to build autonomous and adaptive systems, so systems that don't need high amounts of tuning, but ones that could perhaps adapt themselves through feedback to achieve certain application metrics. Folks talked about the idea of could you actually use transcriptional regulation in a precise way to be able to modulate multiple things inside the cells in a high throughput fashion. So can you very quickly design something that will work and actually do what you want it to do? One of the things that came out, I think, in both the sessions was the challenge of getting consistent performance, especially as you move your transcriptional circuit between chassis. And so can you design circuits that work in multiple contexts as you expect, or if not, can you develop new metrics for trying to characterize how that works? Another thing that came out as an application was, if you can start designing such complex circuits, can you build potentially kill switches or other mechanisms for limiting potential escape of some of these circuits. And finally, I think there was a lively discussion, at least in our group, about whether the correct way of designing synthetic circuits is to build something that's highly orthogonal, using non-native parts, versus trying to repurpose perhaps human or mammalian parts in some way to build a circuit that perhaps is not so wildly different than what you might find inside the cell. So I'll talk on this slide about half, and then I'll turn it over to John. So the application pull that we spent a lot of time on in our group talking about, folks were interested in moving gene therapy beyond kind of simple single gene diseases. So can you identify why you actually need such a complex circuit inside of a cell? Are there some complex diseases where there's some network dysfunction that you'd like to be able to reprogram with the transcriptional circuit? I think there was a lot of interest in exploring epigenetic modifications, how do you do chromatin remodeling, et cetera, using some of these synthetic DNA binding domains and fusing different effectors to them. And then beyond therapeutics, I think we discussed, what are some perhaps lower hanging fruit that we can show, that we can work on to demonstrate the power of these particular circuits, including can you build circuits that are optimizing for biomanufacturing? Are there agriculture applications? Are there diagnostic applications, basic scientific tools? Or very interestingly, can you engineer pets or something like that to live longer or do specific things that they're not naturally able to do? So those are all the different applications we thought about, and I'm going to turn it over now to John.
Thanks, Tim. All right, so the push that we talked about in the sessions were essentially new synthetic binding domains and effector domains. So we're currently obviously working on those, and we need to develop more, as has been discussed. As we just heard, new delivery methods, more efficient methods to deliver into primary cells and other cell types. Delivery of our circuits on different scaffolds. So we talked a bit about human artificial chromosomes and safe harbors, and then how do we deal with the contextual differences in safe harbor sites by either insulation or other means. And then again, we did discuss about how do we model these systems before we put them into the cells. So developing more richer computational modeling systems, and we discussed the potential of a big effort to really model the entire human cell, to be able to look at it in a systems type of way, to perturb one side and see what happens, and to get an idea of what might happen when we actually put our circuits into cells. So the roadblocks. I guess the idea, first of all, is to how do we test these systems in vitro rapidly. Of course, we talked about that this morning, to get more of a predictive analysis of our systems in vitro or in easier to use systems prior to going into mammalian cells. And then choosing single strong modifiers versus weak modulators. And we talked a little bit about this yesterday, about noise level in systems, and do you have more of an aggregate effect of multiple transcription factors at a particular promoter, or is it more desirable to have maybe a single very strong effector? So we didn't solve that problem clearly, but we discussed it. And then standardization, I think Tim mentioned it briefly. So in this type of community where we're creating lots of different parts and we're testing parts in lots of organisms, it's going to be important, we believe, to actually have a clear nomenclature of parts, be able to standardize these parts in a standard set of organisms. So do we have a desired set of cells that we use, that we standardize these things in? Do we go back to an ES or an IPS cell to start with, and then have a standard set of methods to be able to look at these parts and these circuits in these cells? And the other thing was to try to take our linear thoughts of some of these systems and look at it in more of a three-dimensional way. So look at three-dimensional types of circuits and systems, really building toward kind of an entire systems thought about that. And that, I think that'll take a significant amount of stretching how we think, and potentially some bioinformatic help there too. And then, again, Tim mentioned, so the orthogonality versus an integrated design. Do we want to go in with completely synthetic networks to be able to measure and actuate on the stimuli, or do we want to have a point where we're integrating our signaling or transcriptional regulation into the host system, and to be able to affect the signaling pathways within the cell? So that's it. Thank you.